Hello, welcome to my channel Deep Learning Biology. My name is Dr. Vikas Bhardwaj, and within this video of molecular biology class, we are going to discuss about the Avery et al. Experiment. Hopefully you have learned clearly the Griffith experiment of transformation. In this video, we are going to learn Avery et al. Experiment. Now here I just want to tell you, you must be knowing in the year 1928, Frederick Griffith, he did the experiment using Streptococcus pneumoniae. And in the case of Streptococcus pneumoniae, you are going to have two strains, one is R strain, another is the S strain. What Griffith found, R second strain of Streptococcus pneumoniae, they get transformed into S third strain of bacteria. So if you have not watched that lecture, please have a look into that lecture in which I have clearly told you every minute details about Griffith experiment of transformation. So here you can see that R second bacteria, they get transformed into S third bacteria. In front of Griffith, there was a question: what is the transforming agent? What is the transforming agent? Now here, before starting the discussion about Avery et al. Experiment, because Avery et al. Experiment gives the idea, gives the clarity, what is the transforming agent. Before discussing the Avery et al. Experiment, I just want to tell you about the experiment done by Dawson and Sia. Now what they did, they took the S bacteria and they heat that. Now they collected the supernatant, they collected the extract of heat killed S bacteria, they removed the bacterial debris and they collected the extract of heat killed S bacterial cell. Now to this extract, what they did, they added the R bacteria. Now they plated this mixture on a LB agar plate, and they were surprised to see that some of the R bacteria, they get transformed into S bacteria. So this experiment by Dawson and Sia, it was clearly saying that you can perform the experiment in vitro. If you remember the Griffith experiment of transformation, so in that experiment, Griffith performed the transformation within the mouse, but here you can perform the transformation of R bacterial cell into S bacterial cell in vitro experiment. Now here I just want to tell you, in the year 1944, Avery, MacLeod and McCarthy, they did the wonderful experiment which clearly proved what is the transforming agent. So here I just want to tell you, Avery, MacLeod and McCarthy, they used hydrolytic enzymes to identify the transforming agent. How they actually did the experiment, we are going to discuss. Now here, what they did, they took heat killed S cell extract, and they removed lipids and sugar from the heat killed S cell extract. How they did that? They actually add alcohol. By alcohol precipitation, they collected the extract. Now from this mixture, you have removed lipids and sugar. Now within this mixture, you are going to have proteins, RNA and DNA. And when they performed the experiment using this mixture, they also saw that this mixture can also transform R bacterial cell into S bacterial cell. Now what they did, they took the mixture and they treated it with protease. In one side, what they did, they treated it with protease, means they removed the proteins. Now you are going to have only RNA and DNA. To this they added the R bacterial cell, and what they analyzed whether the transformation occurred or not. They found that in presence of RNA, in presence of DNA, the transformation occurred. So in absence of proteins, R bacterial cell, they get transformed into S bacterial cell. Same way they did the experiment. They took this extract and they treated it with RNase. Okay, RNase is a nuclease which will specifically degrade the RNA. So now you are going to have within the mixture proteins and DNA. To this they added the R bacterial cell. Here also they saw the transformation occurred. R bacterial cell, they get transformed into S bacterial cell. So here you can see in presence of proteins, in presence of DNA within the extract, you are going to see the transformation. So in absence of RNA, transformation occurred. So here this is going to tell that RNA is not responsible as the transforming agent. Now they did the experiment in another way. They took the extract and they treated it with DNase. DNase is the nuclease which will specifically degrade the DNA. So here what they saw when they added the R bacterial cell. So here you are going to have the extract which is going to have proteins and RNA. And to this extract, when they added this R bacterial cell, what they found no transformation occurred. So in absence of DNA, R bacterial cell, they did not get transformed into S bacterial cell. So try to think here. In absence of proteins, transformation occurred. In absence of RNA, transformation occurred. In absence of DNA, transformation did not occur. So here you can see in absence of DNA, transformation did not occur, means DNA, it is responsible for the conversion, for the transformation of R bacterial cell to S bacterial cell. So here this experiment was proving DNA is the transforming agent. So here you can see that this experiment clearly proved DNA is the transforming agent. But here I just want to tell you, many scientists at that time, they were speculative about the work done by the Avery et al. Why? The reason was simple, because the enzymes which were being used by Avery et al. In their experiment, they were crude enzymes. They were isolated from animal source. They were not so much pure as like we are going to have today. So many scientists at that time, they were skeptical about the work done by Avery et al. Anyhow, you must have understood the experiment done by Avery et al. It was clearly proving that DNA is the transforming agent. But scientists, they need more evidence. So in the next class, we are going to discuss 1953 experiment done by Hershey and Chase. This experiment, by using bacteriophages, Hershey and Chase, they clearly proved DNA is the genetic material. So in the next class, we are going to discuss Hershey and Chase experiment. Thank you for watching this. Have a nice time.